Our speaker for today is Dr. Lauren Zarzar. She's a professor at Penn State with appointments in the Department of Chemistry and the Department of Material Science and Engineering. Prior to Penn State, Professor Zarzar earned a BA in chemistry and a BS in economics from the University of Pennsylvania, a PhD in chemistry from Harvard, and completed a postdoc at MIT. Some of her recognitions include the Packard Fellowship for Science and Engineering, Sloan Research Fellowship, and Camille Dreyfus Teacher Scholar Award. Dr. Zarzar is also a senior editor of the ACS journal Accounts of Material Research. Her group's research interests include the study of colloidal active matter, laser direct writing of nanomaterials, and microoptics. Thank you so much, Dr. Zarzar, for joining us here today. The stage is all yours now. Right, perfect.
Gonna hide the meeting controls. Okay. All right. Well, it's my pleasure to be here today and share with all of you some of our research, both current and ongoing for the last several years, related to the self-organization and behavior of non-equilibrium active droplets. So my group has really taken a lot of inspiration from how nature moves, organizes, communicates, whether that's between organisms or within organisms. How can we design non-equilibrium motility, organization, pattern formation in synthetic materials, and how can we take inspiration from living systems? So it always helps to start by looking at what nature does. I'm just going to show a few examples. So for example, here is a video of, on the microscopic scale, a white blood cell chasing a bacterium. Obviously, the white blood cell can't see where it's going, but yet it has mechanisms by which to both sense and move in a very functional way on the microscopic scale. And now here's a macroscopic scale example where we have a herd of sheep being herded by sheepdogs. And although each individual sheep appears as a tiny white speck, and you take this aerial view of the herd and how this herd is being funneled through these restrictions and fences, and you can see these beautiful patterns emerge. And it almost looks like a liquid flowing through a constriction. And so what is giving rise to these unique patterns and the behaviors that the sheep is exhibiting? You have both repulsive and retractive and non-reciprocal chasing interactions going on here. And how do those give rise to this large scale behavior? So two very different length scales, but yet built on pairwise interactions between constituents and non-equilibrium motility. In terms of pattern formation, I'd love to give some examples from plants, actually. So there are, across the desert, people have noticed for many, many years the formation of what are called fairy circles. So they're these spontaneously formed empty patches of circles spread very evenly across miles and miles of desert. And people haven't really known. I'm still not sure if there's consensus exactly on what's caused by this, what's causing this. But I think that I'm going to vote for the fact that these holes form actually because the plants are competing for a very scarce resource, which is water. And so when you're competing for a scarce resource, how can you cover more area? You can put holes and you can put patterns into a material. Very much like this plant has evolved to have holes in its leaves, where it's essentially trying to find more light in an environment where light is scarce. So whenever you have an active system, and anything alive, really, you consider an active system a non-equilibrium system, and you have a scarcity of resources, and you're trying to look at how the system uses those resources and self-organizes itself over time and space, I think you're going to find that you're going to give rise to some really interesting organization and patterns. And so all of these themes and examples, while not directly related necessarily to the chemistry that I'm going to be showing you today, serves as a form of inspiration for the kind of systems and patterns that you could imagine evolving. Now, as I mentioned, we are particularly interested in bio-inspired materials, so we're not using anything living. Everything is synthetic, but how can we take inspiration from living systems and apply them to non-living, non-equilibrium systems, particularly synthetic colloidal systems? In our case, we're going to be looking at droplets. And I also just want to point out that throughout the talk today, I'll be showing lots of patterns and behaviors, and I'm going to refer to this as self-organization rather than self-assembly. Now, I think when it comes to colloidal organization, self-assembly is very commonly used to describe how colloids will, for instance, pack in space and interact with each other and form larger structures. And also self-assembly sounds very similar to self-organization. So why aren't these two words interchangeable? And so the way I see it is that self-assembly is an equilibrium process. So for instance, if you put a bunch of monodisperse spheres together and you do evaporative self-assembly on that, you're going to get a very ordered crystalline structure. And this is the most stable form of the organization, so we call it self-assembly. It would be going towards equilibrium. Whereas if you find that you have pattern formation or structure formation out of equilibrium and it's fueled, I'm going to refer to that as self-organization. So for example, you could take the same spheres, but now those spheres only come together and form some organized structure when they're being driven by some form of energy. So there's fuel being expended. And by fueling the system, you form that structure. But if you stop fueling the system, the structure falls apart. In this case, the structure is driven out of equilibrium. It's formed out of equilibrium. I'm going to refer to that as self-organization. So all of the patterns and structures that I show you today, they're all self-organization. When you stop fueling the system, all of the structures fall apart. So the only way to get organization is when you're expending energy. So this is what we're going to be looking at today. Now, what are our active agents? What are the things that are going to be moving? And there are many choices that you could make. The active colloids community is actively growing, and there are many examples of active colloids. The ones that we're going to be using in our work are droplets. And these droplets are solubilizing. So what does that mean? Well, basically, we have a droplet like oil and water. And we do put a surfactant in there. So the fact that the surfactant is above the critical micelle concentration. So there are micelles. And these micelles will actually uptake the oil from the droplet to make what we call oil-filled micelles. And this process of taking the oil from the droplet and putting it into the water and putting it into these oil-filled micelles is called solubilization. So these droplets are solubilizing, and through this process, they are creating gradients of these oil-filled micelles around their surface. Now, why do we care about that? Well, you have these clouds of oil-filled micelles, and if you have some spontaneous polarization in this cloud, well, now you have a concentration gradient of the oil-filled micelles across the surface of the droplet, which, for reasons that are actually still being studied, you can actually translate this concentration gradient of oil-filled micelles to a surface tension gradient or an interfacial tension gradient. So if these oil-filled micelles are chemorepellents, i.e. they raise the interfacial tension where they are of higher concentration, then you would get an interfacial tension gradient like shown on this slide. And due to the Marangoni effect, this interfacial tension gradient will actually cause fluid flow, and this fluid flow will allow the droplets to move. So an active droplet can become self-motile. There's no external applied gradient here. The droplets create their own gradients and respond to them, and they can move around spontaneously in solution like you see here on the slide. Now, in addition to being self-motile, droplets can also be repulsive. So they may not necessarily move spontaneously on their own, but if you put two droplets close together, they will spontaneously move apart from one another. Again, because these oil-filled micelles are a chemorepellent signal and they want to get away. In addition, we've also reported some chemical conditions where droplets can be attractive, which would mean that the droplets, the oil-filled micelles, are actually a chemoattractant. And again, this depends on the chemical makeup of the system, which I'm not going to go into great depth today, but I will be putting references on the slides in case you want to follow up later. So when droplets, oil-filled micelles, are chemoattractants, you flip the interfacial tension gradient. Now the flow goes in the other direction, and droplets want to move towards higher concentrations of the oil-filled micelles. And this means that if you put two droplets within a few body lengths of each other, they will actually come together and form an aggregate. So we've had droplets that can self-propel, droplets that can repel, and also droplets that can attract. So we're building up our toolbox, if you will, of different pairwise interactions that we can design. But additionally, and very importantly, we've added an interaction to this set, which is a non-reciprocal interaction. So a non-reciprocal interaction would be like predator and prey, like the sheepdogs herding the sheep that I showed earlier. This would be the case where you have a predator droplet that is attracted to and is chasing a prey droplet, and the prey droplet wants to get away from the predator. So it's repelled by the predator. How do you generate these sort of interactions? Well, for one, they can only be generated under non-equilibrium conditions. You cannot get a chasing interaction under equilibrium conditions. So everything has to be under non-equilibrium, which we know. But then how do you think about designing the chemistry? How do these droplets interact with each other? How to design this. So this is what we did a few years ago now, where we demonstrated that when you take one droplet that is solubilizing, such as bromopentane, in a non-ionic surfactant like Triton X100, and you mix that in solution with a non-solubilizing droplet. So in this case, we used ethoxy nonofluorobutane. This is a non-solubilizing fluorinated oil that is, the oil is miscible with the bromooctane. So the two oils do mix. You put these two droplets together in a dish in this Triton X100 surfactant, and we see that these droplets will chase each other. The bromooctane is attracted to the fluorinated oil, and the fluorinated oil wants to swim away, but eventually it's caught. So the predator catches the prey. And once the droplets touch, the pair of droplets will continue to move through the solution in a very directional manner due to this non-reciprocal interaction. So what's causing this behavior? We're going to use a relatively simple framework, which is a source-sink pair. Now, on the molecular scale, actually, this can get much more complicated, but conceptually, the idea of using a source-sink pair works well. Now, a source in this case would be the bromooctane, because that's the oil that is solubilizing. It's creating this cloud of oil-filled micelles. And the fluorinated oil is our sink. The fluorinated oil does not create oil-filled micelles. It does not solubilize. However, because these two oils are miscible with each other, and so there is a favorable free energy of mixing, the oil from the oil-filled micelles in the source will get transferred to the sink. So the oil is being shuttled through the water from the source to the sink. And all of this is being facilitated by micelles. So remember, we have these micelles in the solution. They solubilize and uptake the oil, and they can also transport that oil back into the sink droplet. Now, this forward transfer is fast because the source is solubilizing, but the reverse process is very slow because the sink oil, the fluorinated oil, does not solubilize. So you end up having this directional net transfer of oil from the source to the sink. And if we remember that in this case, the oil-filled micelles are a chemorepellent, both droplets want to get away from high concentrations of solubilized oil. This sets up a situation where the source is attracted to the sink and the sink is repelled from the source because both droplets want to get away from those oil-filled micelles. So conceptually, a source-sink framework can explain this chasing behavior. Now, previously, we were very interested in how the different chemistries of oils in the source and the sink impact their behavior. So we looked at all different combinations of droplet oils, but more recently, and what I'm going to be primarily talking about today, is how the behavior of the system doesn't necessarily depend on the chemistry, but more on the self-organization dependent on the size and number of droplets. So we had previously seen some really fun examples of self-organized behavior. I'm just going to let this video play. I think it's one of my favorite videos. These droplets they're the same droplets, bromooctane and the ethyl nonylfluorobutyl ether, but the droplets are different sizes, and you can see how the droplets self-organize and change their patterns dynamically over time. And I always thought that was so fun, and we wanted to learn more about it. So for the rest of the talk, we're keeping the chemistry of the droplets the same, although you can change the chemistry to affect the behavior. And we're going to be looking at how the physical inputs of the system, like the size and number of the droplets, how does that impact the self-organization that we see? Yes, we mentioned that. Okay, so our experimental design. We use the same oils as I was showing you before. So the bromooctane, which is going to be our predator, ethoxy nonylfluorobutane, which is our prey, and Triton X100 surfactant. We use microfluidics to make the droplets. So we use a glass microfluidic chip. It allows us to form monodispersed droplets of precise size of each type of droplet, which we collect. And then when we're ready to look at their interactions, we will then mix those droplets together in a glass bottom chamber that we watch on a microscope. And we take videos, and then we will do analysis on those videos to try to pull out quantitative analysis. So when we mix two populations, we see some behaviors emerge. So I'm going to show you some data where we're looking at number ratio of the droplets of 10. So there's 10 times more bromooctane than ethyl nonylfluorobutyl ether. Ethoxy nonylfluorobutyl ether, excuse me. And the diameter ratio is 0.5, meaning that the predator droplet is twice as large as the prey droplet. The prey is twice as large as the predator. So this is what video. When you mix those populations of droplets, this is what emerges. And we see these flower patterns where you have the prey droplet completely surrounded by the predators. Now, it takes a minute or two for these flowers to form. And then if you wait long enough, we actually see that the flowers fall apart. But for some time period in the middle, approximately 15 to 20 minutes, these flower patterns are relatively stable. They don't exchange droplets. They are pretty static. So we're going to refer to this time period in the middle as a steady state, where we're going to sort of disregard the few minutes that it takes for the organization to form, and we're going to disregard the end where everything falls apart, and we're going to look at the middle. And this is going to be our dynamic steady state. And I'm referring to it, of course, as a dynamic steady state because the droplets are still communicating with each other. In principle, they could still exchange components, exchange droplets between different clusters, but the pattern itself is steady. So the pattern isn't going to change that much. Okay, so we have this dynamic steady state that's forming. And we're going to look at how that dynamic steady state changes as a function of the physical inputs of the system. Now, I just showed you this example where our number ratio was 10 and our diameter ratio was 0.5. So what if we keep the number ratio the same, but we change the diameter ratio? Let's increase our diameter ratio. Well, this is what we see. The pattern itself, i.e. the flower pattern with having the prey in the middle and the predators on the outside, stays the same. But because of geometric considerations where now our predator is much larger, we can't fit as many predators around each prey. So we see that the cluster shape changes a little bit. But actually, maybe this is really not that surprising. The interactions between the droplets are the same, and you still pack all these predators around the prey, just limited by geometry. Okay, so let's change the number ratio now. So we're going to keep the diameter ratio the same, and we're going to change the number ratio. And when we did that, this is the video that we see. So here's the number ratio of 1, diameter ratio of 0.5. So if you think back to the flowers that I was showing you before, that was the same diameter ratio as this, but a number ratio of 10. So all we've done is change the number ratio, and this pattern is so different. I would call these more like chains and loops being formed rather than those flower patterns. So if we were to put some pictures here, I see we go to a number ratio of 1 and then 0.1. We now are forming these chains and loops rather than these close packed flower structures, which is much more interesting to me. I'm trying to think, well, why is this happening? Like, why don't we just form the inverse flower structure, right? Why don't we just simply have now the red predator spheres surrounded by a bunch of blue prey? So if we look at the pattern phase diagram to summarize everything that I've shown you so far, these are the examples that you've seen. When we had the high number ratio of 10, we saw these close packed flower structures. And then when we kept the diameter ratio the same and changed the number ratio, we went from these close packed flowers to these chains and loops. And for the sake of clarity and completeness here, I'm going to fill in the rest of the patterns that I didn't necessarily show you examples of, but this is what they look like. Now, when we look holistically at this pattern phase diagram, there are some really interesting things that emerge. The first observation is that while we got close packing with the excess predators, so the number ratio of 10, we never see close packing when we have a excess of the prey, so the number ratio of 0.1. And the other interesting thing was that these patterns are non-commutative. So you can't simply take two inputs and inverse them and get an inverse structure. So for example, this yellow box here in the 0.5, 0.1 category, the inverse of that would be 2 and 10, right? Just take the inverse, but the pattern doesn't invert, right? So we go from chains to flower patterns when we invert the diameter ratio and invert the size, invert the diameter ratio and invert the number ratio. So the patterns are non-commutative. Why is that? So we wanted to try to understand this, and we believe that it is because of the fact that all of these, largely, these interactions are driven by this non-reciprocal interaction. So if we think about why these patterns are being formed in the first place, let's take a look at the case of the excess predators. So I like to think of this as a situation of you have a bunch of wolves and they're chasing a deer. So you have a bunch of predators, they're chasing prey. Now, once they catch that prey, all of those wolves are going to pack around that deer to share in the meal that they've just caught. And they're going to close pack around their prey. And once there's no more room around the prey, they're going to have some repelled predators that can't quite fit. And that actually describes the pattern that we see for the case of the excess predators. You have this singular prey surrounded by as many predators as can fit, and then the ones that don't fit are going to be repelled. Now let's consider the case where we have excess prey. Well, one wolf can't chase 10 deer all at once. Now, it looks like from our droplets that actually our predators can chase and catch more than one prey. We do see examples where the predator can be attached to maybe 2 or 3 prey without there being another predator around. But certainly, this predator cannot catch 6, 7, 8 prey all at once. So it's really hard for a predator to chase multiple prey at once. And in fact, this is what we see in the droplets. Because of this non-reciprocal interaction, the predators can only go after and catch a minimal number of prey at once. And this gives rise to this loose packing and the fact that we have a lot of escaped prey that don't close pack around the center predator droplet. So we end up with this very loose packing architecture where we have these chains and loops, or if we do see a flower pattern, it's not a close packed flower pattern, and there's going to be space around the predator. Now, we think that this can describe qualitatively all of these patterns that we see. And so it's really interesting because the patterns in this pattern phase diagram, if you will, is all built on pairwise interactions that involve a non-reciprocal interaction, which is only allowable out of equilibrium. So you can't get this under an equilibrium organization condition. And you can only get this out of equilibrium for a driven self-organizing system. Now, for those of you in the audience who may be more theoretically inclined, you can recapitulate a lot of these patterns from a relatively simple dry model. Now, what do I mean by dry? Using these equations, these relationships, we're not taking into account any sort of convective flow. And we're treating the droplets as two-dimensional disks, ascribing them some activity and some mobility, and looking at a concentration gradient of the solubilized oil that decreases at some radial distance from the edge of the predator disk. And when you ascribe these pairwise interactions to this minimal model, and we're using the experiments to inform the values of activity and mobility, but then we're allowing this model to evolve the different patterns. We do see that we can actually recapitulate the general behavior that we see in the experiments. It's not completely reproduced. There are some differences, which might be a result of the fact that, for instance, we're not taking into account convective flow. We're also not taking into account changes in activity and mobility that might be arising as a function of the different inputs like diameter ratio or number ratio. But given a relatively straightforward system applied to a relatively complex three-dimensional droplet system, the fact that we can get these general patterns to emerge, I think is quite interesting. Now I want to come back to the end of life of these patterns. So I began by saying we were going to look at the steady state, and we looked at the steady state, but then I said, well, if you wait long enough, the droplets fall apart. The droplet organizations fall apart. So you have this pattern, but then if you wait long enough, the pattern dissociates. And it's not because the droplets disappeared. The bromooctane is still there. So there's still fuel, if you will, to solubilize. But something's changed where the pattern is no longer stable, and so everything falls apart. So what's happening? So we think that the droplets are falling apart because when we think of this in terms of a source sink framework, our sink is overflowing. So all of this interaction revolves around a non-reciprocal interaction that is driven by the transport of oil from the source to the sink. If your sink does not have an infinite capacity, which it does not, then eventually you're going to fill it up and the interaction is going to weaken. And we can see evidence from this in experiment, where if we look at the center droplet, so the center droplets are fluorinated oil. It has a low refractive index of 1.28. The predator droplets have a high refractive index of 1.45. Now, when we transport oil from the predator to the prey, we raise the refractive index of the prey. You can see that eventually the center droplet becomes invisible. It's still there, but the reason it becomes invisible is because you've put so much bromooctane into it that it actually raises the refractive index of the fluorinated oil, which is the prey, to the refractive index of water. So now the prey droplet has the same refractive index as the water, and that means you can't see the prey anymore. So once we get this index matching, and over time, as the refractive index of the prey goes up, eventually we see that the cluster falls apart. And if you watch that fluorinated oil droplet for longer, we actually see that now it becomes visible again at the 33-minute mark. So some of that bromooctane that it had uptaken, it now releases back into the water. And this change in refractive index, which indicates the transport of oil from the predator to the prey, we see that this can be correlated with the time scales over which the droplets will dissociate from their clusters. So conceptually, what we can think of is that you're weakening this non-reciprocal interaction over time as you're filling up your sink. So you cannot put an infinite amount of the bromooctane into the fluorinated oil. Eventually, there's diminishing returns from this free entropy of mixing. And when those interactions weaken, then your cluster will fall apart. Now, we were wondering, can we change the direction of this non-reciprocal interaction? We weren't able to invert those flower patterns with simply physical changes like diameter ratio and number ratio, but could we use chemistry to change this interaction? And previously, we had shown that if you swap your surfactant, so if you change your Triton X100 to, say, a fluorinated surfactant, you keep the oils the same, you just change your surfactant, that you can, in fact, change the chasing direction. So the surfactant that we had been using, which is Triton X100, it preferentially solubilizes the bromooctane. So the bromooctane was our predator. But if we change our surfactant to a fluorinated surfactant, this fluorinated surfactant now preferentially solubilizes the ethoxy nonylfluorobutane, and it does not solubilize well the bromooctane. So effectively, by changing the surfactant, we've changed the designation of predator and prey. And when we look at the pairwise interactions in this video, that is what we see. Now, the interaction seems to be a little bit weaker. The droplets don't move as quickly. They have shorter ranged interactions, but we do see that the prey droplets from the Triton X100, which were the fluorinated oil, they're the lighter colored droplets in this video, they are now our predators, and they are chasing the darker colored droplets, which are the bromooctane. Now, if we take this new surfactant and we apply the same thinking that we had previously done with the Triton X100 and look at different number ratios, different diameter ratios, what do we see? And I'm going to just show part of the phase diagram here, where we have a diameter ratio of 1 and a number ratio going from 0.1 to 10. This was the phase diagram you already saw in the Triton X100. This was an example of our transition from the loose packing to the close packing flowers. we see these flowers. We're going to look at the same part of the phase diagram, but now our surfactant is a fluorosurfactant. And so the chasing direction is reversed. And what do we see? The patterns are now reversed. The designation of predator and prey has been swapped, and these patterns are now swapped. So the flower pattern that existed where you had excess bromooctane when you had the Triton X100 surfactant, we now see flower patterns with the predator fluorinated oil in the fluorosurfactant being on the outside. So we can generate the exact same qualitative patterns, but we've just swapped the designation of what's the predator and what's the prey. So if you wanted to make an inverse flower pattern with the bromooctane in the middle and the fluorinated oil on the outside, we could never achieve that in the Triton X100 by simply changing the physical inputs, but we could achieve that by changing to a fluorosurfactant. So there are certain patterns that you can generate, certain organizations that you can create by changing the physical inputs. And there are certain changes that you can make by changing the chemical inputs. And they don't necessarily always overlap. They're very synergistic. And so trying to understand what different physical and chemical inputs can do to your system in terms of the self-organized patterns, I find quite fascinating. So what happens if we have a third oil? We had a predator and a prey. But what if this prey was actually a predator to a third oil? What would happen? Like, could we introduce a third droplet here and make a chain of chasing interactions? Well, we had to think carefully about what this third droplet would be if we wanted to, say, make a pattern that looks like this. Right? You have the blue droplets chasing the red droplets and the red droplets chasing the yellow. What would be the yellow? Well, we needed a droplet that could mix with the bromooctane because you have to have a source sink pair, but doesn't mix very well with the fluorinated oils. So we chose halogenated hexadecane. And I'll say the only reason we're using halogenated oils is simply because they're denser than water. It just makes it convenient. There's not really a chemical reason for it. But these hexadecane oils, they mix with the bromooctane, but they're very immiscible with the fluorinated oils. So this yellow droplet would not be a good sink for the blue. And if we leave both surfactants in there, so now we have to have a system where both the fluorinated oil and the bromooctane can both be predators. So we have to use both surfactants. So we have a system with 3 oils and 2 surfactants. We mix them all up, and we do generate the patterns that we intended. So I'd show this as an example of you can extend this framework to many different situations, and you're going to evolve much more complex patterns. And I'll also say that I'm showing you examples of the clusters that we intended, but as you can see, like in this bottom one, you're getting some loops. So it's not to imply that every cluster in this mixture looks like this. You get a mixture of different structures. And so something that could be investigated in the future is, for instance, what does the distribution look like? How do those differently organized clusters move and interact with each other? That's something that we haven't looked at yet. Now, for the last few minutes of the talk, I want to show some examples of self-organization where we keep the chemistry of the droplets the same, but now instead of looking at 2 separate droplets, we're going to fuse them together. So we're going to keep chasing single emulsion droplets, but now make them into a Janus droplet where we have an oil-oil interface. How do these Janus droplets self-organize? Now, for these examples, I'm going to be using slightly different oils because bromooctane, as I had used throughout the rest of the talk, Bromooctane is fully miscible with the ethoxy nonofluorobutane. So if you were to try to make a Janus droplet out of them, it wouldn't work very well because the oils would just mix. So we're going to use iododecane, because iododecane can still form a chasing pair with the ethoxy nonofluorobutane, but it's not fully miscible. So when the droplets are in a Janus droplet, they don't fully mix. So we make Janus droplets of these two oils. Again, we use microfluidics. We can very precisely control the volume ratio of the two oils in the droplet. And when we look at the behavior of these Janus droplets in the Triton X100, we see that the droplets, they move in the general direction that we expect. The iododecane is the predator and the fluorinated oil is the prey. So in the Janus droplet, that is the direction that they move. It's the same direction as in the chasing droplets. But the Janus droplets actually move much faster for this specific example. They're moving about an order of magnitude faster than the chasing single emulsion droplets. And I also really like this video because you can see how the droplets interact with each other's trails. They don't ever crash into each other. They will actually interact with the trails left behind by the other droplets, which we've color coded here. So the trail age changes over time, and that's indicated by the color. And it's just a reminder that as these droplets are moving, they're leaving behind a trail of these oil-filled micelles. You can kind of think of it like the jet trail behind a plane. And over time, those oil-filled micelles, which are a repulsive chemical signal, but over time, those oil-filled micelles will diffuse away. And so that signal disappears. But on short time scales, those trails can actually be quite a repulsive signal to the other droplets. So the trails can allow a way for the droplets to communicate with each other, and this can also impact their self-organization. Now, if you have certain chemistries of droplets that really don't want to mix together, but they're fused together in a Janus droplet, you can actually reverse the chasing direction. So you can get a Janus droplet that moves in the forward direction, but kind of similarly to how we switched the surfactant and we made the droplets move the other way. You can do something similar with Janus droplets. Not only by using surfactant, but you can also play with the miscibility of these Janus droplets and get them to swim in the reverse direction as well. So you have some of the same tunability that you do with the chasing emulsion droplets, but now we have them in a Janus droplet. Now, what do these Janus droplets do in terms of their collective behavior? Well, one fun thing that we found is that you put a pretty high number density of droplets together, they will spontaneously form these spinning clusters. And these clusters can have 3, 4, 5 or more droplets in them. And the direction that they spin depends on the orientation of the Janus droplets in the cluster. And if we look at the flows around these droplets, we can actually see they look a little bit like a propeller spinning, and the droplets are drawing in solution from above them and spitting it out the sides. And we think that one of the driving forces for this organization is, if you will, a scarcity of resource, which is their surfactant, right? In order for these droplets to solubilize, they need oil, they need micelles that are not filled with oil. They need empty micelles. And most of those empty micelles are actually located in the solution above the droplets. So remember, these droplets are sitting on a glass cover slip. In this video, they're surrounded by a lot of other droplets that are all solubilizing. So there's a lot of oil-filled micelles in their immediate vicinity. Most of the fresh surfactant without oil in it is above them. And somehow, we're not entirely sure, but somehow their confluence of driving forces that cause these droplets to come together in these clusters. And by spinning, they can actually much more effectively draw solution in from above them and shoot them out the side. It's like a reverse tornado. And this will allow the droplet to actually solubilize faster and better because it can access that fresh surfactant much more efficiently. And that fresh surfactant is a scarce resource when you're surrounded by a bunch of other droplets that are all solubilizing and using that up at the same time. So I think it's a nice corollary in a way to the plants. The droplets are organizing as a way of using their resources at a different efficiency. So you can also use applied gradients, in this case, thermal gradients, to control droplet behavior. This is something that we had not done before. So all of the previous examples, organized behavior was all spontaneous. There's no externally applied field. But here, if we shine a laser at the droplet, so the red dot is a near IR laser, it raises the temperature at that location by not very much, probably just a few degrees. But this temperature gradient is enough to drive organization of the Janus droplets with this thermal gradient. Thermal gradients can have quite a few effects on these active droplets. Of course, temperature changes directly affect surface tensions. They also affect solubilization kinetics, as well as the mixing of the two oils in the droplets. So there are a lot of factors that temperature can influence. And these Janus droplets appear very sensitive to temperature gradients. So you can drive the droplets to organized and orient using a laser. If you scan the laser back and forth, you can change where the droplets are looking, right? So they're looking towards the heat source. So you can use various forms of thermal gradients. You could also think about using electric fields, or use an electric field to generate a thermal gradient in order to orient these droplets and move them in certain places. Okay, so with that, I'm going to conclude some takeaways. Non-reciprocal chemotactic interactions between droplets, which we can conceptually think about in terms of a source sink framework. These non-reciprocal interactions give rise to all sorts of self-organized patterns, both in chasing single emulsion droplets, but also in Janus droplets. And these behaviors and these organizations are different than those that you could imagine in an equilibrium situation, an equilibrium self-assembly. These patterns are different because they're all driven by a non-reciprocal chasing interaction. In addition to the chemistry of the inputs, so the surfactant, as well as the choice of oils, you can also change the number ratio, diameter ratio, in order to tune the organization. So we have these different handles by which to manipulate the structure, and we're starting to understand the synergies between both the chemical handles as well as the physical handles. And this simple, well, relatively simple framework where we primarily were looking at just two oils and one surfactant, so chemically pretty simplistic, it can be extended. You can add different oils, you can use mixtures of surfactants, you could think about integrating this with a reaction going on, something that's going to shape those chemical gradients in a much more complex, maybe time-dependent way. And you can extend this framework to more complex systems to potentially create much more hierarchical organizations that maybe change over time. And I have many, many people to thank and acknowledge. Of course, without the inputs and hard work of the many students, postdocs, and collaborators that I've had, none of this work would have been possible, and I just get the pleasure of sharing it with you. I've highlighted a number of papers in this talk, and I also want to make sure to thank the funding organizations that have also made this work possible. So thank you all for your attention. I'd be very happy to take any questions.
Thank you so much, Dr. Zarzar, for such an insightful talk. Now, as our moderators collate question from the chat box and the Q&A, we would be running a short poll for our audience. Shubhangi, please go ahead. All right. So you can see three questions on your screen. The first one being, are you attending ACS Science Talks for the first time? The second, how did you hear about this session of ACS Science Talks? And lastly, what brought you to this session of ACS Science Talks? You have got 10 seconds to answer these questions, so requesting you all to start the poll. All right, thank you so much. Can we please have the results? Right, so here we are. A majority of them pointed that they are not attending science talks for the first time. Thank you for that. And most of them heard about ACS Science Talk session through email, and they have come due to scientific curiosity. Thanks everyone for participating in this poll. Reshmi and Jinsi, over to you.
Thank you, Neha. Um, Thank you again, Professor Sir, for this exciting talk. It was so beautiful to see how the different processes in nature has inspired such interesting research. Without further delay, let me quickly jump into the questions. We have received quite a number of questions on different applications, potential applications of these droplets and their interactions. So could you please share your thoughts on what are the potential industrial applications or possible applications as nanomotors or in drug delivery?
Mm-hmm. Yes, so as you actually just mentioned, nanomotors and micromotors have been proposed, for example, for active cargo delivery, potentially for sort of therapeutics. Probably not these droplets. I don't think these droplets would hold up very well in the body for that particular purpose, but you could imagine a solid particle, this, for instance, driven by a reaction, or a lipid nanoparticle that can be motile and maybe shuttle a drug or some other therapeutic to a specific place in the body. There have been some studies looking at how active droplets, for instance, could maybe at a solid surface, you could imagine for surface cleaning or for collecting pollutants. We've done some work on droplet optics. So the Janus droplets, for example, if you can position them in different places, you can orient them. We also have mechanisms to change the structure of the Janus droplet, so the curvature of the interface between the two oils, that can be modified. And you can use the droplets as a lens. So it could be a tunable or responsive optical system as well. Those are just a few examples. I will say these are very fundamental studies. And some of the things that we're learning just in general about how emulsions behave under non-equilibrium conditions at a fundamental level can have implications for applications and use of emulsions that doesn't necessarily have any tie to their active behavior. But things like partitioning, solubilization kinetics, mechanisms, all of those are important for just understanding how emulsion droplets work. And emulsion droplets are used in so many commercial applications, from paints and coatings to foods and personal care product formulations. They're all made with emulsion. So the more that we can understand about non-equilibrium droplets and emulsions, the better.
Thank you, Professor. And we have a few more questions about the role of temperature. So one question is, at what temperature are these experiments carried out?
So all of the experiments I showed, with the exception of the one with the laser where there was a temperature gradient, all of the experiments are carried out at room temperature. So we mostly out of convenience. And then for the situation where we had the temperature gradient, we didn't directly measure the temperature with the laser, but we did apply thermal gradients in another manner laterally, and with only a few degree difference over many, many millimeters, we could generate directed motion and organization of the droplets.
And there is a related question. So do you think that there is a role for this temperature in the organization of these droplets?
So when we're not applying any sort of temperature gradient, I don't think that temperature is playing too big of a role. We do have, I mean, these droplets are all in a dish where there is a top surface most of the time that's exposed to air. So you can get background convective flows just from having that set up, which you could, tie back to thermal gradients and buoyancy, et cetera. So you have these background convective flows, but they're weak compared to the chemotactic interactions that you're generating between the droplets. So the droplets, the strength of the droplet interactions with each other is stronger than the background convective flows that you might get from, say, a vertical temperature gradient.
All right. So you were talking about like all the possibilities to control or tune the self-organization of the droplets. So is it possible to control the movement of these droplets?
Control the movement.
Movement and its kinetics.
So the speed of the droplets, their motion can be controlled through a number of parameters. One is their chemistry. Two is the surfactant concentration. So within a range, if you increase the surfactant concentration, you're increasing the solubilization kinetics, you can increase the speed within a range. There's a limit to that. You can also use temperature. So for the Janus droplets, when we did more controlled experiments with temperature, we haven't done this with the single motion droplets, but for the Janus droplets, you could increase the temperature and increase their speed again within a limit, which could be, again, just be due to faster solubilization kinetics. You're basically expending your fuel faster, you're driving everything faster, and that makes the droplets move faster. But there's a limit to all of this. You cannot simply just keep increasing the temperature and expect things to be increasing in speed. But the Janus droplets, we've had those move and up to 200, 250 micrometers per second, so they can get quite fast.
All right. Yeah, as we are running out of time, I would request you to share your final remarks and thoughts before we close the session.
Yeah. Well, final remarks. Well, thank you, of course, for having me here. Really enjoyed being able to share with you some of this work. If anybody has any follow-up questions that we did not get to, please feel free to send me an email. I didn't put my email on this slide, but you can easily find it by Googling me. And also, as was mentioned at the beginning, I'm an editor for Accounts of Materials Research. So I would love to have contributions from those of you in the audience. If you'd like to learn more about that, please don't hesitate to reach out to me. So thank you very much.